Welcome everybody to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you Anna Huttenlocher.
It's good.
No, go ahead.
Huttenlocher or Huttenlocher?
Huttenlocher or Huttenlocher. You do the German too.
Huttenlocher, yeah.
She is a physician scientist and has appointments both in pediatrics and in the Department of Medical Microbiology and Immunology. She's going to be here talking with us about going forward in reverse. How does inflammation resolve? I'm going to ask her the 5 questions. Anna, how do you pronounce your name?
Huttenlocher or Huttenlocher?
So good. That's not everybody gets that. Good ask that. Now, where were you born?
I was born in Boston, Massachusetts.
And where'd you go to high school?
I went to high school in Chicago, Illinois.
Oh, where?
In the south side of Chicago.
And where'd you go for your undergrad and what did you study?
Oberlin College. I studied biology, but I'm also a violist. So I played in the Oberlin Conservatory Orchestra. So I like music too.
So what did you have against the violin?
Oh, well, this is kind of interesting, actually. So I started on violin, and then my grandfather had an old viola from the 1700s, and he gave it to me, and I switched instruments because of being given such a special viola.
This is why we ask these questions.
Yeah.
That's great. And then where'd you go for your advanced degrees?
I went to Harvard Medical School.
And did you do both your medical, your MD?
I'm just an MD only, but then I focused on science after.
Good. And did you do any postdocs or anything like
Yeah, I
that?
did a postdoc at UCSF.
Okay. And then when did you come to Madison?
99.
Right. And tonight, I'm looking forward to your talk about inflammation. I'm a plant pathologist and plants don't get inflamed.
Oh, I'm not sure. Do they? Everything gets inflamed.
Well, now I'm just going to walk away in humiliation. So look at this. Whoa, I'm embarrassed. Let's find out if plants do get inflamed. I don't know. You're a horticulturalist.
I bet they do.
Okay, well, I'm batting 0 for 3.
When a
here.
microbe hits them, they get inflamed.
Okay. So this is really good. Elizabeth, you'll get to edit this all out. Would you please join me in welcoming Anna Huttenlocher to Wednesday Night Lab?
Well, thank you so much for that introduction. That's really fun. I welcome questions throughout the talk. In fact, to me, it is more fun if people ask questions, and this is more of a dialogue than a presentation. And the other reason that I want you to ask questions is I'm going to show you a bunch of movies. So we're very lucky in that, oops, yeah, it is on. Okay, could I move it up further?
Yeah.
I always have this problem because I have a quiet voice, maybe even further. Okay, is that better? Is that better?
Yeah,
All right.
just talk as loud.
Okay, I'll talk as loud as I can. Yeah, I have, my grandmother was an opera singer and she had this very loud voice and mine is quiet. So I didn't inherit her vocal cords. So anyways, yes. So please ask questions and interrupt throughout the presentation because it's much more fun in that way. And as I said, I'm going to show you a bunch of movies, but let me just tell you a little bit about kind of my training and background. So I'm a physician and I went through the first year of medical school and then decided, you know, I'm tired of memorizing and I really want to think about what's going on with patients. So I started to do science between my first and second year of medical school. And then from then on, knew that I really loved science. And so for me, I've had a job where I've been 20% clinical and 80% research. And I see children who have autoimmune diseases and immune disorders. So disorders like lupus, various forms of arthritis, or different types of immune dysregulation. And so they have really been kind of the stimulus behind what I'm going to tell you, and really the research that we do has been inspired by patients that we see in clinic. And I also just want to highlight the idea that you can look at a very simple model organism, in this case, a zebrafish, and the zebrafish are transparent. And the advantage of zebrafish is that you can watch things and you can learn absolutely new things about biology just by watching. And then when you make these observations, people can go into mice or think about it in humans. And in the case of the story I'm going to tell you tonight, it really was backed up by mouse and human data. So this is the strength of the zebrafish model. So I'm going to be talking about how inflammation starts and stops with a focus on this question of how it resolves. Can you guys name some examples of inflammation? Wound healing, exactly. So you get a scratch, that's acute inflammation. It gets infected, that is kind of acute, but it can become more chronic. Oftentimes it gets cleared by the immune system. Sometimes you require a little bit more, some antibiotics, but absolutely wound healing. And then you can have chronic wounds with diabetes, for example, wounds that don't heal. And we're very interested in those sorts of questions. Other examples of inflammation.
Sprained ankle.
Sprained ankle, right. So some sort of injury, which can actually go on for weeks or even longer. So it's not really acute, like, for example, a very small cut. It becomes longer in duration.
Gum disease.
Gum disease, yes. So gingivitis is a big issue that involves inflammation and involves the immune cells I'm going to be talking about a lot tonight, which are neutrophils. Absolutely. Someone else said that. Arthritis. Yep. Arthritis is another example.
What about long COVID?
Long COVID, absolutely. Or acute COVID. So an aberrant immune response, for example, the kids who get MIS-C, who we've been seeing in the hospital quite a bit, although interestingly, we have not been seeing them lately. So something's changed in the pandemic where we initially saw these children with acute systemic inflammation associated with COVID, and they required medicines that would dampen the immune response, like steroids and IVIG. So yes, COVID is an example. So in various infections, other examples, cardiovascular disease, cancer, Alzheimer's, neurodegenerative disease. These are all examples where inflammation can drive disease. And as we're learning more and more about many of these disorders, we're learning how important it is to understand inflammation more broadly. So do you all know what types of cells are a part of inflammation? Can you?
Leukocyte.
Leukocytes. So leukocytes are a general term usually used to describe cells of the innate immune response, like neutrophils and macrophages. So absolutely, leukocytes, and they're the first responders to tissue damage. T cells. So that's part of the adaptive immune response. So when you think about, with COVID, you initially have the activation of the innate immune response, and then subsequently, you hope you have a nice, robust T cell response, antibody production that helps protect against the infection longer term, one hopes. But it's very good at evading the immune system. That's one of the challenges. Other cells. So actually, many cells of our body are involved in the immune system. So epithelial cells, our skin, they make cytokines and inflammatory mediators. Fibroblasts, which are part of the connective tissue, they also make inflammatory mediators that influence how neutrophils, macrophages, T cells behave. So these are all parts of the inflammatory response. And this is a fun aspect of it, is you can't just say it's just the cells of the immune, traditionally what you think of as the immune system, but it's a bigger part of the puzzle, because these cells in the tissue microenvironment regulate what the immune cells do. So they're critical parts of this whole process. So one thing as a physician scientist is many of the questions that I started out with when I started my lab were driven by patients I saw in the clinic. And so this is a child that I saw when I was still in training at UCSF, and I was a fellow. And this child presented with chronic fevers, skin rash, and many of these patients actually have central nervous system inflammation that can lead to deafness and developmental delay. And they also have joint swelling. And this is a biopsy of the skin. And all of these purple cells in here, the nuclei, are of neutrophils. So these are the cells I just described, these primary cells of the innate immune response. And these patients get treated with medicines that dampen the immune response. And we had some interesting results with this patient, and I'll describe it later, where it puzzled us about the relationship between how cells of the immune system move, how they migrate, and the relationship between the start and stop of inflammation. So again, just highlighting how important it is in terms of the patients we see, the questions that they raise, and then ultimately our goal to try to translate what we find back to the clinic. So the neutrophil, as I mentioned, so it's really a balance with these cells. If you have too much or too little, it's a problem. So here you can see a very classic movie from the 1950s. This is a neutrophil in hot pursuit of a bacteria. And the neutrophil is pushing through the red blood cells. Should we lower the lights in the front? Would that help? Yeah, I'm not sure. Maybe it's here. Lights. Hidden stage.
Can't see it. We can see it.
You can see it okay? Okay. Anyways, you think it's okay? Okay, good. Anyways, so you can see this neutrophil in hot pursuit of a bacteria pushing through red blood cells. Finally, it will reach the bacteria, it will phagocytose it, and this is absolutely required for host defense. So if you don't have functional neutrophils, you cannot survive. So kids who are born with neutrophils that are not functional, but get treated with a bone marrow transplant. So they are absolutely essential for life. But the problem with these cells is they are just very good at going into tissues and causing all kinds of havoc. So they will lead to tissue damage, increase the risk for autoimmunity, and contribute to chronic inflammation. So it's really a balance of having enough neutrophils there, but not too many neutrophils there. So that's one of the themes that will come out today. So these neutrophils are really good at migrating. So this is a system where we've collaborated with bioengineers, and I think there have been past talks on microfluidics. So this is microfluidics with the BB laboratory. And what we've done is there's an endothelial lumen that has neutrophils in it. And then we've put aspergillus, and these aspergillus fungi really like the endothelial lumen, and they will form what are called these hyphae. You can see them, these long structures. And these hyphae are very stimulatory to neutrophils. So the neutrophils are first just moving randomly. Then a neutrophil attacks a hyphae, and one of the neutrophil actually releases chemokine. And this is a type of motility called swarming. Once a neutrophil gets activated, it releases chemokines, inflammatory mediators that recruit in more neutrophils, and you get this really strong immune response. And these cells are capable of chewing up these hyphae. Now, when fungal infections really take hold, they're very hard to treat. So actually, something I'll talk to you at the very end of the talk today is we're working to engineer neutrophils using stem cells to make them better at fighting things like infection or having anti-tumor functions. So if you can have, in fact, what we've already found is that neutrophils that come from these human inducible pluripotent stem cells are actually more effective at fighting fungal infection. So it's actually pretty exciting that maybe this will help patients who don't respond to antifungals like amphotericin. And this is particularly a problem with patients who are on chemotherapy and are neutropenic, either due to chemotherapy or for other reasons. And I'd like to acknowledge Laurel Hind, who is a former postdoc in the lab, who now is running her own lab at University of Colorado, Boulder. So this is one of the pleasures of being a scientist, is being involved in training really talented people who then go on and run their own research programs. So we are interested in what recruits these cells to either sterile tissue damage, so without an infection, versus infection. And we wrote a review article a number of years ago that highlights kind of the complexity of this. And I don't expect you to go into any detail, but it just tells you that there are phases of this inflammatory response. First, a neutrophil is responding to pretty simple signals at the wound site, what we call DAMPs. Then what happens is there's a kind of a activation immune response where you get release of chemokines, and neutrophils are recruited to these chemokines. And then you get the amplification, that swarming that I showed you with the hyphae, where the neutrophils become super activated and they're recruiting in more immune cells. And then if you have an infection, even different signals are developed where the neutrophils will come and they'll stay, and some of them will die, and they do something called mitosis, where they release their nuclear material, and that helps trap microbes and is involved in the clearing process. So we're interested in what distinguishes neutrophil response to a wound without infection and to infection, and then what signals promote resolution of inflammation. So why zebrafish? Why do we use zebrafish? So what do you think about this movie? Absolutely. Isn't it incredible how well you can see this? So this is a zebrafish larvae that is 72 hours post-fertilization. And you can see that at this stage, what do you see that's different between neutrophils and macrophages? So the top ones are neutrophils, the bottom ones are macrophages. Speed. And so neutrophils are much faster. They get to the damage really quickly. What else do you notice that's different? The organization is very different. So neutrophils, a bunch of them are trapped in the bone marrow. And then they don't come out of that bone marrow, it's called CHT in the zebrafish, unless there's damage or infection. So they're not very motile there, but the second there's damage or infection, they move out of that spot and move very quickly to the damage. Whereas macrophages are just slower and all over the place. So they do this, what we call surveillance migration. So they're just checking everything out. And if there's a problem, they'll recruit in neutrophils to help them. So it's really elegant when you think about it. And at this stage of development in the zebrafish, they don't have an adaptive, they don't have T cells yet. So I have a postdoc who's studying T cells, the much later stage, but this stage, they only have this innate immune response. And that's really the focus for today's talk, is this innate immune response. And what's really cool is that we can, for example, in this case, put something into the ear, where we just injected, in this case, we just damaged it with a needle. And let me see if I can move this. And then we can also cut off the tail. And what you can see is that, oh, actually in this case, sorry, this was infection. So there's Pseudomonas in the ear. And we cut off the tail, and that's a sterile injury. And what do you see? What's happening in this top movie? Can you guys see it well enough? So the neutrophils are going both to the wound and to the Pseudomonas, the bacteria that's in the ear. And then we take a drug, and this inhibits a signaling intermediate called reactive oxygen species. And we inhibit reactive oxygen species, and what do you see? What changes? They're only going to the bacteria. So this highlights the idea, and this is the really cool thing about zebrafish, is that you can stick a drug in the water and see how it differentially affects the response to bacteria as compared to sterile tissue damage. And what's very interesting too is in response to infection, you get even more neutrophils recruited. So in that case, the reactive oxygen species seems to dampen the inflammatory response where it's required for them to get to sterile injury. So this is where there's so many layers of this regulation, the signaling, that has a different effect on neutrophil recruitment to infection as compared to sterile injury. So the other thing that's nice about zebrafish is we can model human disease in zebrafish. And so I'm going to give you an example where a little baby came into the clinics at UW and we found a single gene defect in the baby. And because the innate immune system is highly conserved in zebrafish, we could introduce that mutation into the zebrafish and recapitulate the human disease. And so in this case, you can see this is the patient neutrophils that we use microfluidics to check the motility. And here's a control. And what do you see is the difference? Right, so patient neutrophils are really slow. So this patient presented with recurrent infections and actually got large abscess formation. And the neutrophils just did not migrate at all. So this suggested a disease called leukocyte adhesion deficiency to us. And we found out the patient had a dominant inhibitory mutation of a signaling protein called RAC2 that is absolutely essential for neutrophils' ability to migrate. So when this mutation is expressed, it inhibits the function of the gene that exists that's normal in the baby. And so we expressed this in the zebrafish. And what we found is that it affects cell motility. So this just tells you, kind of summarizes cell motility, that you have these chemokines that I mentioned, these inflammatory mediators. They get recognized by receptors, and this activates this RAC, which is mutated in the patient, and this is important for regulating the ability of cells to migrate. So without that, they don't migrate normally. But what's really cool about this, and this is kind of the fun thing where you can be goofy in the lab, is that if we take a photoactivatable form of this protein, or we can activate it, and we repeatedly activate it with light, we can actually control where the neutrophils move in tissues in an animal. In this case, spell the word RAC. Now, everyone in the lab was like, why didn't you spell your name? Why didn't you spell, anyway, so this went into the paper. This shows that this pathway is sufficient to induce the ability of neutrophils to migrate, but is it required? So here you can see a control neutrophil migrating, and we're using some actin probes here. And when we express this mutation, the neutrophils are just stuck. They can't move. They're not polarized. The actin is not polarized like in the control. So this absolutely blocks the ability of the neutrophils to move. And then we look to see what happens with infection. So we know the baby, if they get infection, they can't survive. And so what happens with infection is what you can see, unless you put them on antibiotics that can control it. But ultimately, that will not work. You can see neutrophils don't come to the site of infection in the ear, and the zebrafish die of the infection. So this shows you, not only can we use the fish to watch things, we can use drugs to try new drugs that might affect inflammation, but we can also model human diseases. So we have a weird patient come in, we can model it in zebrafish and say, okay, this mutation is causing the disease. So it is a very powerful system. So back to this patient that I mentioned to you. Oh yeah.
Do you usually then, because this sounds like an autosomal dominant condition, do you check parents just to make sure?
Yes.
Yeah.
So it was a new mutation. So with autosomal dominant, like a severe mutation like this, the parents wouldn't have been able to survive. Yeah. So it was a de novo mutation. Yeah. But very good question. And so years ago, when I saw this patient at UCSF, I drove out to the patient's house. They live in rural California, went to their farmhouse, drew her blood, and went and looked to see how her neutrophils migrated. And I thought, with all this inflammation in the tissues, her neutrophils would migrate better. But what I found, and this puzzled me, is that her neutrophils didn't migrate as well. Not as badly as that patient I just showed you that had severe infections, but they definitely had impaired migration. So this raised the question, what is the relationship between neutrophil migration and inflammation? And can you learn new things in the fish system just by watching? So our lab was the first lab, including a lab in England at the same time, that generated zebrafish where you can see the neutrophils. They're green, they're expressing green fluorescent protein. And so under a neutrophil specific promoter, and here's a wound, and tell me what you're seeing. What do you guys see?
They go to it and then they go back up
Yes,
towards whatever.
that's the blood vessel. Absolutely. So they go to the wound and then they go back to the blood vessel. Well, and as a medical student, you learn that neutrophils go into sites of tissue damage and they die. They don't leave. So you watching this, you're going, no, 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 they are leaving. This was very controversial. And so I went to every mouse person I could find and said, you got to try this in mice. Otherwise, people are not going to believe it. And it took over a decade later for them to show that this actually happens in mice. So we can also do something pretty nifty where we take the first 5 neutrophils that come to a wound site and we shine a light on them and switch their color from green to magenta or red. And you can see that the neutrophils do this funny thing where they're very indecisive. Yeah, they're going back and forth and they can't decide, which makes perfect sense, right? If their infection happens at the wound, you want the neutrophils close by. You want them to be in the vicinity. But if that wound signal really dissipates and the signal at the blood vessel is stronger than the wound signal, then they'll leave, they'll go back. So this is exactly the questions now we're really, and we and others are working on to try to dissect and figure out what's going on. And if you look at the blood vessel, you can see the neutrophils first coming out of the blood vessel, and then they're going back in and they're poking in and out a lot. And so there is evidence now from mouse models that this poking in and out is involved in alerting the immune system. It tells the T cells to get activated and it kind of activates the immune response. So not only is this a process of resolution, so we can track where these neutrophils go, and a lot of them end up going back to the hematopoietic tissue, where they will eventually die. Some of them will go to other tissues, and then they're activated neutrophils ready to respond to the next problem faster. So they are changed neutrophils. So just to summarize, so how does neutrophil inflammation resolve? The traditional thought is that neutrophils go into sites of tissue damage and they die. And what we found is neutrophils can, in some contexts, go into sites of tissue damage and leave. And I'm not going to show you this, but if there's infection, most of them will die. Oh, question.
So how do they get in and out of the blood vessel?
Yeah, so there's been a lot of work to understand the transmigration process of attaching to the endothelium. The neutrophils will roll and they have these surface receptors called integrins and selectins that bind to the endothelial surface and they'll move out of the tissue. There's less known about that reverse movement, but there's growing evidence to suggest that specific chemokines and pathways are involved in that reverse movement. So good question. But the blood vessel and the neutrophil and how they talk to each other is a very interesting area. And so this process has an important role in disease implications. So there's a phenomenon called ARDS, Acute Respiratory Distress Syndrome. And so when I was speaking to audiences years ago before the mouse data, the pulmonologists, the lung doctors would come to me and say, oh, this explains it. This is what's happening. So what happens is a patient has a lot of trauma, for example, or they have something called pancreatitis, so they have inflammation in their pancreas. And the neutrophils go in there, they become activated, and then those neutrophils can actually go to the lung, where they cause a lot of damage and problems and lead to this acute respiratory distress syndrome. So now that we understand that process, can we use drugs to block this process of reverse migration? So this is a question that's interesting. Now, there's also been recent studies showing that neutrophils will associate with tumor cells as they're metastasizing in the circulation. And so if you have neutrophils with those tumor cells, the prognosis tends to be worse. So that's another whole area that is just really in their infancy, but pretty exciting. An additional one is patients with lupus. So I don't know if any of you know people with lupus, but if you go out into the sun, it can lead to inflammation in your whole body. And so a recent paper came out that showed that when you stimulate a mouse with UV light, neutrophils come to that site in a lupus model, and then they leave by reverse migration and they go to the kidney. And these neutrophils are activated. They release their nuclear material, and then this helps lead to the production of autoantibodies, anti-nuclear antibodies. So there's all these sorts of things that are really quite interesting that's coming out. Now, this is sort of a fun one that was done by a former postdoc in the lab who now has her own lab at Albert Einstein University, Sofia de Oliveira. And what she did is she fed fish either normal diets or high-fat diets. And what you can see is that neutrophils and the pink nuclei come to the wound. And what do you see with a high-fat diet? A lot more neutrophils. So it does make you think about the next time you go get Big Macs every day for a week. So a much bigger inflammatory response. And what Sophia found is that if she went and took neutrophils that were in the liver, so when you have a high-fat diet, you get inflammation in your liver. And she photoconverted those neutrophils in the liver. In the control diet, none of the neutrophils came from the liver, but with the high-fat diet, a bunch of them are photoconverted and came from the liver. So this is, again, an example of how you can use this model to probe conditions such as how diet influences the immune response. And Sophia now has her own research group, and she's pursuing these questions. Yeah, so she is interested in high sugar as well, but it's certainly a very interesting question about how having a very high sugar diet might influence some of these things. Earlier today, we were celebrating one of my PhD students defending her prelim, and we went to the terrace, and I had one of those big bowls of ice cream. And to be honest, I hadn't had one of those for a long time. And afterwards, I was just like, woo, the sugar high, and then so who knows what my neutrophils were doing in that case. So we use all sorts of techniques to be able to figure out how gene expression changes in neutrophils and macrophages with tissue damage. And so I'm sure you guys have heard about RNA-seq probably, have you? So it's looking at gene expression, single cell RNA-seq, where we look at gene expression changes in single cells. And then CRISPR, where we go and we knock out things by targeting the pathways that change in different conditions. So we were interested in genes that change in neutrophils and macrophages with sterile wounding without infection. And then we also were interested in this idea of cancer as an unhealed wound. So a lot of people look at cancer as a wound that just keeps being inflamed and it is unhealed. So in this case, a former graduate student, Ruth Housewright, and then a whole team in the lab fished for new neutrophil intrinsic genes doing an unbiased screen where they did what we call trap RNA-seq. So we express a protein specifically in neutrophils or macrophages that binds to RNA, and then we can pull down those actively translating genes, and we can look to see how gene expression changes with a wound. And one gene came out that there was not much known about, although there was a study in mice that showed that this seems to be an important pathway in response to ischemic damage to the heart. So the mice do better when they have myeloid-derived growth factor than if they don't in response to having like a heart attack. And so, so we were curious about this, and this just shows you, so we used CRISPR and we knocked the gene out in zebrafish. And you can see when we injured the zebrafish, you can see neutrophils and macrophages being recruited to the wound. And when you don't have this myeloid-derived growth factor, and the side view shows it very nicely, there are many more neutrophils that come to the wound. So this is a pathway that inhibits neutrophil inflammation and sterile injury and not infection. So this is something we were particularly interested in. We didn't want to block neutrophils and infection because that would make people get bad infections. We wanted to find a target that blocked neutrophils just in sterile injury. And so we're quite excited about this. We submitted a patent to WRF, and we're interested in this as a potential therapeutic. So what happens if we just inject the protein into the ear of zebrafish? So this, if we inject just media into the ear, you get neutrophils, these white dots. If we have this protein, the myeloid-derived growth factor protein, then you don't see nearly as many neutrophils, and this is quantified here on this side. And if we have the knockout, you can see there are many neutrophils in the ear, and this is rescued by the protein. So, as I said, we're really excited about this as a potential new inhibitor of neutrophil inflammation that blocks what we think might be bad neutrophil inflammation, where wounds don't heal as well, but doesn't affect it when you need the neutrophils for infection. So this gives you an example of the kind of work that we can do in the zebrafish model, where we go from imaging, you make an observation, to doing gene expression analysis, and you see new genes that are changed in different types of inflammation, to taking CRISPR and knocking out the gene and seeing what the impact is. And then doing some protein purification where you put the protein back and see what happens. So it just is sort of the process of discovery and how powerful zebrafish can be. So I just wanted to talk briefly about this idea of cancer as an unhealed wound, where neutrophils and macrophages are both recruited to the tumor cells. And neutrophils tend to come in and come out of the tumor cells, whereas macrophages come and they stay, and you can see they just hug the cancer cells. They just, kind of surround the cancer cells with these protrusions. So we are very interested in this idea of neutrophils as a target to treat cancer. So neutrophils are the first cells to get into tissues. They're so good at getting into even dense tumor cells. So you guys have probably heard about CAR T cells as an approach to treat cancer. But one of the big challenges is that T cells aren't as good at getting into solid tumors. So this CAR T cell is really good for treating, hematopoietic malignancies. So where it's in the blood, but not as good at treating solid tumors like a lung cancer, for example. And so we're interested in the fact that neutrophils can be either anti-tumor or pro-tumor, and trying to figure out the genetic pathways, which many groups are working on this question of gene pathways that make a neutrophil anti-tumor, because this could be very powerful in terms of treatment because they're so good at getting into, for example, tumors, because it's a type of damaged tissue. So we did find that myeloid-derived growth factor is also elevated in immune cells in the tumor microenvironment. And we found that if we deplete myeloid-derived growth factor, it affects the ability of those tumor cells to invade. So this is something that we're very interested in doing. This is just showing you some gene expression changes that happen in the tumor cells themselves, where you get a lot of upregulation of genes, and in the neutrophils, where you get up and down regulation of genes in response to exposure to tumor cells. So this just, again, goes back to the fact that we can go to these observations and then go back and image. In this case, the macrophages are in green, the neutrophils are in blue, and the tumor cells are in magenta. And with time, when these immune cells are interacting with the tumor cells, many of the tumor cells die. So the magenta, the pink, is lost over time. So I'd like to end, this is a little bit technical, but I think it raises some interesting points at the power of the gene expression analysis that we can do at this point. So what I was showing you so far was gene expression changes in tissues. So like in neutrophils from the whole animal or macrophages from the whole animal. But we can also just look at single cells. And this is the amazing, just here in our biotech, this building, you can send your sample and they can look at how gene expression is changing in single cells. And this allows you, for example, if you take a tumor, you can see all the cells that are in a tumor, and you can see how different tumors influences gene expression. In this case, this is a tissue damage model. We burned the zebrafish, and you can see that we can see all these different types of neutrophils and macrophages by this color coding here, and the change with the wound. And this is quantified here. You can see, actually, interestingly, many genes are upregulated in neutrophils with this type of damage, but the macrophage genes are downregulated. So it just tells you how complicated all of this is. And we can go and we can choose one gene that we are interested in called interleukin-6, which is a cytokine. It's an inflammatory mediator. And we can look to see how that changes. And we can look to see what happens with its receptor. And what's very interesting is we found that IL-6, which is the ligand, is only expressed in macrophages. So the macrophages make the IL-6, and the neutrophils have the receptor. So these cells are together in a damaged tissue, and the macrophages say, oh, here's some IL-6, and the neutrophil has the receptor, and that can influence neutrophil behavior. And so we went and we knocked out this IL-6, and what you can see, and actually you can tell me what you see, neutrophils are in pink, here's the control, and here's the knockout. What do you see? The neutrophils do not go to the wound. The macrophages go to the wound just fine. So this is a beautiful example where you go from the gene expression data saying, oh, the macrophages don't have the receptor, it doesn't change, or not much of the receptor. The neutrophils have the receptor, but the macrophages are making the IL-6, and that you get rid of the IL-6 receptor, and neutrophils can't come normally to a wound site. Now, I just want to highlight, and in fact, it's kind of blocked here on the bottom, that actually there are drugs that we use all the time in our patients with arthritis, with vasculitis, which is inflammation of the blood vessel, that block the IL-6 receptor. So this is a very good drug, but it's also a drug that can have side effects because it's blocking this key cell from getting into sites of tissue damage. So I'd just like to end with this idea that we can go from all of this work, imaging to screening to gene expression analysis, and go back to humans. And this is just this idea that we can engineer neutrophils, and I'm not going to go through all this, to actually be better at fighting infection. Like I mentioned to you, the iPS-derived neutrophils are really good at fighting fungal infection. We can also engineer them potentially to be anti-tumor. And so that is work that we've been doing with Igor Slutkin, which has been very exciting, where we've been making neutrophils from iPS cells that are compatible with human treatment. So they're called GMP compatible neutrophils. So we're hoping that this gets into the clinic and can help patients directly. And just want to end with that concept that critical to all of this is this balance of having not too much neutrophil inflammation, but not too little neutrophil inflammation, and having the right type of neutrophils coming in to sites of tissue damage so that it can actually do their work properly. And I'd just like to end and say I work with a great group of people. Here are some of the recent graduates and postdocs, many of whom are now running their own research laboratories. And working at Wisconsin has been amazing. It's such a collaborative, great work environment. And as was said early, that my lab was at the terrace enjoying this beautiful evening. So thank you so much for being here, and I'm happy to answer any more questions.
If you have a question, I'll put you talking face against the microphone with the red duct tape.
Certainly the pictures you showed, the zebrafish and neutrophils and lactate is beautiful. And visual models to show what types of neutrophils. You also mentioned that you want to advance this model to mice and humans. Can you talk a little bit about how that's done in terms of replicating the information you need from the zebrafish?
Yeah, so example, I mean, this is where scientific conferences are so important. So I presented this work at scientific conferences all over the world and went to mice people who had the ability to sterilely injure a mouse and photoconvert the neutrophils and track where they're going. So they found that they go to the lung and then they go to the bone marrow. So they reverse migrate out of the sites of sterile injury. So, that's the beauty of collaboration in science. So once you know that and you can identify some drugs, then you can test them in fish, you can test them in mice. And if they're drugs that have already been approved in humans, then you could test them in humans, right? If it's not, that's pharma, it takes years. It's a very long process in terms of drug development. So, yeah.
What I was trying to ask was, how do you monitor those neutrophils in the mice and humans? Do it so clearly with the zebrafish, but you can't imagine how you photo watching these neutrophils in these other models.
Right, so mice, it is a challenge, but they open up the mice and you stick the microscope lens into the mouse and you can watch the neutrophils just in the tissue and you can photoconvert them. While the mouse is alive. And then you go and you sacrifice the mice and you take the liver or the lung or the bone marrow. And because they've been photoconverted, you can do something called flow cytometry, and you can look for the photoconverted neutrophils. And they found that they go to the lung where sometimes they actually cause damage, as I told you in the case of lung inflammation with ARDS. Ultimately, they go to the bone marrow, which is a controlled environment where they can die and they're not as damaging. There are all these macrophages there that can gobble them up.
Have you used any other models like Hecker-Cho cells, or are they useful in terms of inflammation, just to see if you can repeat what you saw in zebrafish, which may lend itself much easier to high throughput drug screening too?
Yeah, so we've been using iPS-derived neutrophils for that, for higher throughput analysis or knocking out pathways in a human system. So many years ago, I worked with CHO cells for quite a bit, and that was to do basic biochemistry and looking at signaling pathways and some regulation of adhesion. But they don't move like neutrophils. They can't do this chemotaxis and this rapid migration. So you really have to work with a neutrophil. And I think our best is to either get them from humans, or the iPS-derived neutrophils.
Another question about cancer being, most cancers also cause a hypercoagulability state. Is there any association between, inflammation and coagulability?
Yeah, so that's a very good question. So COVID, you get a lot of inflammation and you get a hypercoagulable state that can be problematic. That's why a lot of patients are put on anticoagulants. So megakaryocytes are, the big generators of neutrophils, and they're very interesting, platelets, which are involved in coagulation. And they are an interesting kind of immune regulated cell. So yes, there is a deep connection between the production of platelets and inflammation.
And if you read the two that are in the chat from Mark and respond to those, that would be great.
Okay, can you discuss the staged foreign body reaction, the role of neutrophils and macrophages based on your current knowledge? So if you have a foreign body, in general, if macrophages are close by, they'll go there very quickly. And depending on the size of the foreign body, they will try to engulf it and clear it. They have organelles called lysosomes that have all these enzymes that can be used to degrade some foreign bodies. But it can often be a problem where the foreign body is too big and you just get this immune response. If macrophages aren't close by, neutrophils are often the first ones to get there, and then they'll recruit in macrophages. So hopefully that answered your question on that one. Then the other question was, several of your slides show the presence of ROS, reactive oxygen species. Can you discuss ROS's role in inflammation and tissue damage? So reactive oxygen species are very important mediators of inflammation. But the interesting thing about ROS is it can also be involved in signaling that can help to resolve inflammation. So if you remember the movie I showed early on, where we had an inhibitor of ROS and we got more neutrophils at an infection site, it's this kind of conundrum that some pathways are involved both with the start of inflammation and also the ending of it. And this is what makes it very hard for some drug development. So reactive oxygen species and neutrophils and macrophages are absolutely important for killing specific microbes. In fact, there are humans that are deficient in their reactive oxygen species signaling pathways who end up developing specific infections that are quite devastating. And so they have to be treated to counter this. So.
Can you remind me what reactive oxygen species are? So two seems pretty reactive to me, but
so one of them is hydrogen peroxide.
Okay.
Yeah, so that would be, and then there are other reactive oxygen intermediates. But for the purposes of the wound environment, we think a lot about hydrogen peroxide.
Need that?
Okay.
Thank you. Yes, so exciting. In your wildest dreams, what are 2 or 3 human outcomes that would be fueled by this research? Disease remediations or your wildest dreams?
So my wildest dreams is a drug like myeloid-derived growth factor, or a drug that influences inflammation and sterile injury, but not infection, can be used to treat patients with arthritis and other inflammatory diseases where neutrophils cause tissue damage without a risk of infection, this is as a battle for us as physicians, is that we use drugs that are great, but they have risks associated with them. So that would be one wildest dream, is come up with a drug. My other wildest dream is we could take these neutrophils and engineer them to cure solid tumors like pancreatic cancer that we can't help currently. That's my other wild history.
I have a daughter with psoriatic arthritis, so I understand the dilemma.
Yeah. But good question. I think, I love basic mechanisms. Like I love thinking about the basic biology, but ultimately it does get back for me as a physician to how this could translate.
And the question I want to ask is, what percentage of impact do immune suppression drugs have on neutrophils?
So what percentage?
Well, yeah, percentage of impact do immune suppression drugs.
Yeah, a lot of immunosuppressive drugs lead to neutropenia. So in the case of chemotherapy drugs, almost all of them lead to neutropenia. And that is one of the devastating consequences of chemotherapy is patients become neutropenic and they often have to stop the chemotherapy because they develop fever and neutropenia. And this is a real problem. So, yeah.
And there's another one for Mark.
He says, are you aware that oxide nanoparticles have been at high density found in the pancreas of 2 diabetics surrounded by inflammatory cells? The largest source, is toothpaste. Interesting. Maybe we shouldn't be brushing our teeth. That's interesting. I am not aware of that. I'll have to ponder that one. Huh. That's interesting. Maybe we should be using less toothpaste.
What I was interested in is that there's more and more of a link between infections and cancer. And you are differentiating the type of inflammation that occurs with infection from cancer inflammation. Is there a time when that changes in terms of the type of infection that occurs? For instance, like an HPV infection, cervical cancer. Are we looking at, is there a change in what the inflammatory response is after that infection produces a cancer response?
Yeah, no, that's a very good question. So, HPV, various GI issues, you end up having chronic neutrophil inflammation, and chronic neutrophil inflammation can lead to the development of cancer. So you're absolutely correct that chronic inflammation can lead to cancer, and that's a very interesting question. There's also some work that's suggesting that some microbes can lead to activation of the immune response that might be beneficial for the immune system fighting the cancer, because as we know, you get a tumor cell and the first thing to get rid of it is actually your immune system. So there is that kind of dichotomy that is also interesting. But in the classic sense, you get an infection, chronic inflammation, and this leads to an increased risk of cancer.
And Mark has the citation up there for the titanium oxide.
Yeah, that's super interesting. Huh. Yeah, that is why you don't swallow it. But I do like brushing my teeth.
I have a question about vaccines and how they would have an impact on neutrophil formation or modifying neutrophil formation.
Yeah, so vaccines traditionally activate the adaptive immune response. So it is interesting. I mean, for example, the COVID vaccine, when you get acutely sick right after the vaccine, that is the innate immune response. So there is a component of innate immune activation, and that is probably what makes people sick right away after vaccination. But ultimately, the success of a vaccine is going to be how it's activating the adaptive immune response. I'm less familiar with the work on the innate immune response and vaccine responsiveness. So I guess yeah.
Any other questions? I have one. Was that viola made by Mr. Stradivarius?
No. Actually, it was made, my family is German, and it was made by, some local instrument maker in a village in Germany, and it kept getting passed down. So it's pretty cool.
Yes, it is.
Yeah. Yeah.
So it's 300 years old?
Yeah.
You still play it?
Yep. Has a really nice mellow sound. It's very, yeah.
We got it sometime when we need another Untitled Lab.
Yeah, I can play some viola.
Great. If there are no other questions, thank you very much.